All right. Hey there, everybody. Welcome to this recorded lecture. It's too bad that we had technological glitches that is making me have to re-record this all by myself, but that's the way it goes sometimes. So this lecture won't be very interactive, unfortunately, because I can't use my pen using this recording software that I'm using. I can't use my pen to write on the screen, so I'll only be able to point at things. And so you're going to have to take notes very, closely on what I'm saying sometimes. So just make sure you take really good notes. I won't be sort of writing on my slides to help you to know when to take notes. But without further ado, let's get into this. So the digestive system is our second to last lecture. I'm going to get through all these slides all in one video, and then it'll be up to you to watch this video, take good notes, and send me questions by email, okay? And that'll be the way that we'll get you up to speed on this. All right, let's get going. So the first part here is just going to be sort of general functions of the digestive system, define some terminology. I'm not going to review the anatomy of the digestive system. That was in the homework that you completed, homework 16. So I'm going to assume that you're good on the digestive system anatomy. And then what we're going to do is get into the second part of the lecture, where we're basically going to imagine what you had last to eat, maybe for lunch or for breakfast or whatever it was. Imagine that food, that mixed meal. We're going to follow the food and talk about what happens in the mouth, what happens in the esophagus, what happens in the stomach, what happens in the small intestine, what happens in the large intestine. And that's where we're going to leave the story at that point. We're not going to go to the rectum and talk about the pooping and all that kind of stuff. That's just not the way we're going to roll. So we're going to spend our time talking about how the food is kind of manhandled by your digestive system, and the enzymes that are put into play to break down those huge macromolecules that you put into your mouth into small little monomer components that can then be absorbed into your blood. Movement from the lumen of the digestive system into the blood is called absorption. Don't get that mixed up with reabsorption, which is what we learned about in the nephron. All right, so let's get going on this. So just to define some terminology, there's this nice little cartoon figure in your textbook, figure 21.2. It's showing the idea here that we are going to be ingesting some sort of food molecules, big, huge, complex biomolecules from the plants and animal products that we eat. Well, we can't absorb these food molecules, these massive molecules, directly into the blood. They need to go through a lot of physical and chemical digestion. And that's mostly going to be handled by enzymes, because remember, a lot of the biomolecules that we would ingest are all held together by covalent bonds, those very strong bonds that we learned about earlier in the class. And maybe you have to go back and review a little bit of Unit 1 stuff. When we first started talking about biomolecules, I'll say this now, I'll probably say it again later. You may have to go back and review the basics of biomolecules, how they're constructed, their monomer forms, their polymer forms, in order to be successful on the final exam with these digestive system questions. So that's Unit 1, Chapter 2, if you have to go back and review your notes. There's nothing wrong with spending a little bit of time going backwards to Unit 1, Chapter 2. It'll be time well spent to get a good solid refresher on biomolecules. But we eat these huge biomolecules and we need to digest them down to smaller, nearly to monomer level of structure. Some biomolecules must get all the way down to the monomer level of structure. Those are the carbohydrates. But others, like lipids and proteins, need to be digested down to monomer or near monomer levels of structure. And at that point, they can be absorbed across the walls of the intestine. Like this might be the small intestine or the large intestine, cuboidal epithelial cells. So we're going to be talking about the apical membrane. This would be the apical membrane of these cells, faces the lumen of the digestive tract, and then the basolateral membrane. We'll be talking about carrier proteins and channels at work on the apical membrane and the basolateral membrane in order to get these small building block molecules into the blood so that your body can use them. They can be dispersed all over your body, stored, or used immediately. We'll talk a little bit about motility, moving the food from one compartment to the next. Certainly, it's a linear digestive tract. You don't want any backtracking, going in the wrong direction, or anything sitting still for too long. And so motility we'll talk a little bit about as well. Secretion is a term that we've heard before when we were talking about the urinary system, but it means something very different here in the digestive system. So don't get this term secretion mixed up with what we learned about in the urinary system. Same term, different functions, okay? So secretion in this case is going to be these cells that line the walls of the digestive tract here in blue are going to release some of their contents into the lumen of the digestive system. These would be exocrine secretions like hydrochloric acid, digestive enzymes, bicarbonate to neutralize the acidic contents coming from the stomach, for example, mucus to keep everything soupy and goopy and sloshing around in there. So exocrine secretions is what this arrow is meant to represent. This arrow here going into the blood, this is representing endocrine secretions. There are hormones that are involved in coordinating the events happening in various parts of the digestive tract. So this arrow right here is representing endocrine, and also bicarbonate is going to be put into the blood. I just mentioned bicarbonate over here, bicarbonate ions, but bicarbonate ions are also going to be put into the blood as well. And then this arrow going from, say, this cell to this cell, that's implying paracrine signaling. Remember, paracrine signaling we learned about earlier in the class, back in Unit 2, Chapter 6, if you need to go back and review that. But paracrine signaling is when one cell releases signal molecules that influence the function of nearby cells. So that would be like paracrine signaling, okay? So those are some of the important features of the digestive system. We're not really going to talk about storage or elimination of the poo at the end. Like I say, we're going to spend our time with these first four. All right, so these functions are now defined on this slide here. There's various kinds of motility. There's, of course, ingestion, putting food into the mouth. Chewing it up, mixing it with saliva is sort of the next step. That's the technical term for that is mastication. Got to swallow it, of course. The technical term of that for that is deglutition. Just read the slide. These are the two that we'll talk probably about the most: peristalsis and segmentation, or segmental contractions. Peristalsis is sort of a squeezing action, a wave of squeezing that propels a bolus of food or a bolus of chyme from one area to the next. Like from the esophagus, from the top of the esophagus down plop into the stomach, or from one part of the small intestine to another. And then segmentation or segmental contractions is a mixing kind of action to keep the food and the enzymes and the mucus all mixed up together. So segmental contractions or segmentation is a mixing process. I told you about those secretions are either going to be paracrine, endocrine, or exocrine. We'll come back to that later on. The digestion portion is the breaking down of huge macromolecules into smaller building block molecules that can then be absorbed. Trans-Epithelial transport into the blood. There's also paracellular transport to get nutrients from the lumen of the digestive system into the blood. But we're going to focus on the more difficult parts of this, the trans-epithelial transport. That's the absorption bit right there that I'm talking about. And then, like I said, don't get this term absorption mixed up with reabsorption that happens in the kidneys. Two separate processes, two separate words, so don't get them mixed up. And then, like I say, we're not going to spend a whole lot of time on that last little bit. This slide is showing what is the difference between peristalsis and segmental contractions or segmentation. This word here and this phrase here are the same thing, okay? So peristalsis is a wave-like squeezing of smooth muscle cells that propels a bolus of food, a clump of chewed and moistened food, from one place to the next. And so this area is contracting and shoving the bolus a little bit further forward. And then a few seconds later, this area here that was relaxed and accepting the food now will squeeze. Now will squeeze and propel it a little bit further forward. So it's a wave-like squeezing. This area will squeeze, then this area will squeeze, then this area, then this area, then this area, etc., to propel the food from one place to another, like from your mouth through the esophagus down into the stomach is an example of that. Segmental contractions is a mixing action. This happens a lot in the stomach. It also happens in the small intestine to keep the food mixed with the enzyme so that chemical reactions can happen easily and quickly. And so here what we've got is, just imagine this as a section of the small intestine where we've got these different food components like rice and corn and maybe guacamole and beans, all sort of separated. Well, we want to get this all mixed up and homogenated. Well, the idea here is that this section of smooth muscle cells will contract. And then adjacent sections here and here will relax. And then a few seconds later, the area that was contracting will relax. And the areas that were relaxed will contract. And so this is a mixing action. It gets all the foods and the enzymes and the mucus all mixed up so that it's very well homogenized. It also ensures that these food molecules here, as they're being digested by enzymes, stay in contact with the epithelial cells that line the walls, this pink area here, line the walls of the small intestine, so that membrane transporters like carrier proteins and channels can do their job to move food molecules from the lumen of the small intestine out so that they can get picked up by the blood out here. All right? You got to keep that food in contact with those carrier proteins. They can't reach out with like a grappling hook or anything like that and grab onto glucose or grab onto amino acids. They got to grab onto them at their binding sites. So the food needs to stay in contact with the epithelial cells that make up the walls there, and segmental contractions help with that. So it's important for you to refresh your memory of the biomolecule categories that we've been talking about throughout this entire course. And so I give you a handout, a little worksheet on Canvas adjacent near where these slide handouts are for this lecture. And so I want to just review those very quickly right now. So the most important biomolecules that we ingest for fuel would be fats or lipids, carbohydrates, and proteins. And in this worksheet, those are shown down here in blue, pink, and yellow. You can sort of ignore the nucleotides and the nucleic acids for now because we really don't talk about them all that much in this lecture. Yes, you do ingest them. When you eat plant and animal cells, you acquire their DNA. You digest their DNA and RNA and all that kind of stuff. But we're not going to talk about that. We're going to focus on these three biomolecule categories over here because they are the most crucial for as energy energy biomolecules for your body. Also structural too. Taking in amino acids will help with your own protein synthesis in your cells. So just mostly spend your time down here on the bottom of this figure, the handout that I put on Canvas. You don't need to worry about this top part up here. I've actually thought about just sort of cropping it out, but haven't at this point. So remember that for fats or lipids, the polymer form is called a triglyceride. And then the point is when we eat these, we need enzymes to break triglycerides down, back down into their monomer form, back down into either fatty acids or monoglycerides. That's going to be the idea, that there's enzymes that are built to break the ester bonds that hold these three fatty acids to that glycerol backbone. And that's shown over here, sort of in the yellow here. Glycerol joins with a fatty acid via what are called ester bonds, a type of covalent bond, to form a monoglyceride if it's only one fatty acid. Or if you put two fatty acids on a glycerol, it's called a diglyceride. Three fatty acids maxes out the glycerol and forms a triglyceride. Carbohydrates, their storage form is either glycogen or starch. Glycogen in animals, starch in glycogen in animals, starch in plants. And so through the process of digestion, these huge, massive carbohydrates, these polysaccharides, need to be broken down into monosaccharides, glucose, fructose, or galactose. They're glycosidic bonds that hold these polymers together need to be broken by enzymes. And that's what's being shown down here. Glycogen and starch, the two main polysaccharides that we eat, need to be digested down to the level of monosaccharides. Cellulose is also an important polysaccharide that we eat that's also known as dietary fiber. And we'll talk about a little bit about the importance of having adequate cellulose or dietary fiber in your diet to keep a healthy diet. We don't digest this. We don't get any energy out of it, but it does help with various digestive processes. And we'll also talk about its utility in sort of slowing down the absorption of cholesterol and also simple sugars in our last lecture. So that's carbohydrates. And so then with proteins or polypeptides are held together by what are called peptide bonds. And the building block molecule are called amino acids. There are 20 amino acids. There are the essential ones and the non-essential ones. And remember these 4 levels of structure. Once amino acids start to link up with each other and form polymers, there's the primary level of structure, the amino acid sequence. Then that starts to fold into secondary structures like alpha helices and beta pleated sheets. And then eventually the protein takes on a 3-dimensional tertiary structure, which is either a globular protein or a fibrous protein. And then when multiple subunits need to come together, that's the quaternary level of structure, like hemoglobin is a quaternary level protein. So that's just a quick refresher, and this worksheet is meant to be only a minimal refresher. If you have to go back to the Unit 1 notes, Chapter 2, just to refresh your memory on these critical biomolecule categories, that would be time well spent. It wouldn't be a waste. And then this figure is another one from your textbook. I'm just sort of repeating a little bit of what I've already said here. Some of the exocrine secretions that go into the gastrointestinal tract would be enzymes, hydrochloric acid, bicarbonate, and mucus. And then the regulation of this business is going to be via hormones that might stimulate one's appetite or suppress one's appetite. And then paracrine signals are important for local regulation of processes within the digestive tract. And then if you come over here, this figure is showing again the law of mass balance when it comes to the digestive system. The digestive system is an open system. It's open here, it's open down here at the wazoo, and so the law of mass balance is in effect here. The idea here is that the amount of substances that come into the digestive system needs to be equaled with what goes out of the digestive system. And so how do we bring things into the digestive system? Well, certainly ingestion of food and drink. And it's amazing to me that 2 liters of food and drink here taken into the body requires another 7 liters of secretions by the digestive system. Really sort of shows you the amount of energy that would need to be devoted just to digesting your food. So for a total of 9 liters of input. And then if we come over here, the vast majority of those biomolecules, either that we eat or that are secreted, are going to be absorbed back into the blood. And only just a tiny little bit of this is going to be excreted as feces. So very, very little is actually lost from the system as waste. The vast majority of what you eat and drink is going, if it's quality biomolecules, good healthy diet, will be absorbed into the body. I think that's pretty darned amazing. All right, so let's get into the real meat and potatoes, pun intended, of this lecture, where we're going to follow the food from the mouth all the way through the large intestine. Check my time here. Yeah, I'm doing good on time. All right, so this is a sort of a summary overview figure, 21.6. It is going to take this figure takes pretty much the same approach that I'm going to take. We're going to start up in the mouth and then follow the food down the esophagus. We're going to talk about what happens in the oral cavity in terms of secretion, digestion, absorption, and motility. And then we'll move on to the stomach. Things get pretty complicated down there, but we'll hit all of those different points as well. The small intestine is where the vast majority of your digestion and absorption is going to happen. So the small intestine is super, super important to the digestive process. And then the large intestine is just sort of a victory lap, basically, for your food. Everything that wasn't absorbed and digested and absorbed here by the small intestine, there will be a little bit more processing that happens as it moves through the large intestine. But the large intestine is a lot kind of analogous to the collecting duct. It's your last opportunity to take back into the body or take into the body important water, ions, nutrients, that kind of stuff before it leaves out the old back door down here. So this is kind of a nice summary overview figure. It's not all the information that you would need to know, but it's a darn good amount of the information that you'll need to know. All right? So good to check maybe at some point during your study. We finally get to talk now, a little bit at least, about the enteric nervous system. This is a figure from earlier in the class, figure 8.1, back when we were just getting our feet wet with the nervous system. And so the enteric nervous system is a separate neural net outside of the brain and the spinal cord, the central nervous system. A separate neural network that is really just autonomously in control of the digestive tract. Yes, it does get some input from the autonomic efferent pathways like parasympathetic input to it, but it does largely stand alone and separate from the central nervous system. And so we'll talk a little bit about the neural control of the digestive system here. Now there's also hormonal influence on digestion as well. And so there's a table 21.1 that I had you look at for some questions in homework 16. And we're not doing any feedback loops in this unit. I've already said that. So we're not going to get into really strict details about the cells, the stimuli that cause the release of these hormones and the receptors for these hormones and the target cells and all that kind of stuff, because we're not doing feedback loops right now. We're just going to hopefully end the course as a nice, smooth, soft landing without any feedback loops. So make sure, though, I will be asking you questions about these hormones that regulate digestion. So when you're studying for the final exam, make sure you study the questions from homework 16, and you will see that target this table, and you will see very similar questions to those on the final exam. Okay? But we're not going to tear it apart and really unpack it at any great level of detail. We just don't have the time left. All right, so let's start in the mouth. That's where the journey begins. Certainly that's where you're going to ingest food, you're going to chew it up, and then you're going to swallow it. But before you swallow it, there are salivary secretions from the salivary glands. And so there are two important enzymes that are in your saliva, along with a lot of liquid, just to kind of moisten the food, get it all sort of ready to easily go down your esophagus. And so salivary amylase, you've heard of this one before. If you maybe remember Lab 3, when we were learning about enzyme modulation by temperature and pH and various chemical factors. If you have to go back, you can always go back and look at Lab 3 if you want to refresh your memory. Amylase begins the digestion of complex carbohydrates like starch and glycogen down into simpler sugars, down into like oligosaccharides, disaccharides, maybe even some monosaccharides as well. And so that we've heard about before, and here it is again. This is one of the important enzymes that's released by the salivary glands. There's also salivary lipase, and only just a little bit of it is going to be released by the salivary glands. And so we can officially say that carbohydrate and fat digestion begins in the mouth because of these enzymes being released and mixing with your food as you chew it up and as it travels down the esophagus. So a little bit of chemical digestion occurs for carbohydrates and fats, but only minimal fat digestion. Because fats are non-polar, they don't like to mix with the aqueous saliva, so fats just sort of glob to other fats and make like big fat boulders at this point. And so these enzymes like salivary lipase can only nibble at the edges of those big fat boulders. And so there's only a little bit of digestion of fats at this point. Fats will need to wait until they get to the small intestine to really start their digestion, because they need to be emulsified. They need to be made soluble in water, and that's what an emulsifier does. Bile is that emulsifier, and we'll get back to that later on. There is a little bit of absorption, no biomolecule absorption, at least not for energy purposes for the most part, in the mouth. The only place where you can get absorption in the mouth is sublingual, under the tongue. And so this is an ideal delivery method for some drugs, important other medications, like, for example, D-glucose or dextrose. This could be important for somebody who has hypoglycemia. It's a kind of a dextrose gel that can be squirted under the tongue. Quick absorption to help bring somebody's blood glucose levels up. Or various drugs that you would like to have in the blood as quick as possible. Antihypertensives, blood pressure medication, antipsychotics, analgesics. Those are all medications that it would be ideal to have them to have a very fast effect. And then THC and CBD. THC is tetrahydrocannabinol. That's the psychoactive compound in marijuana. And CBD is cannabidiol. That's the non-psychoactive compound, or one of the main ones in marijuana. Everybody seems to think that CBD oil is like the cure-all for everything these days. I'm unconvinced, but I'll be a skeptic to most things. But this is one of the reasons why marijuana edibles can give you a bit of a high rather than having to smoke it. You can get that. Not that I would know, but from what I hear, this is the reason why these marijuana edibles that maybe you've heard of before, or maybe had before, can have such a quick effect on the body, is that there can be absorption underneath the tongue there. And the benefits of a drug, for example, if you ever invent a drug, I highly recommend that you design it to be delivered sublingually, is that it doesn't face the harsh digestive process further along. And so your substance, your drug, doesn't need to have significant protection around it from the harsh acidic environment of the stomach and the more alkaline environment of the small intestine. And it can also have a very fast effect on body function, because as soon as it goes in the mouth and gets across into the blood underneath the tongue, it can have an effect on the body. And certainly we don't store food in the mouth at all, really. You chew it, you swallow it. We're not chipmunks or anything like that. Elimination, under most circumstances, you don't spit your food back out, so we don't really have to worry about those features. So now, this bolus of food, think of maybe you had a sandwich for lunch, or maybe you had some French toast for breakfast or something like that. That bite of food that you've chewed, mixed with those initial enzymes and saliva, needs to get transported from the mouth down to the stomach. Well, it goes through the esophagus. But not much really happens there other than motility. This is where peristalsis occurs, where you get this wave-like squeezing motion, forcing this bolus of food down towards the stomach. And so this squeezing at one section, and then the next, propels the food down to the stomach. Digestion could continue from what started in the mouth, but the esophagus does not really contribute to anything directly. It doesn't release any new enzymes into the digestive tract. So think of the esophagus just like a muscular tunnel that squeezes the bolus of food down, plop, into the stomach. And so now we move into the stomach, and this is where things start to get a little bit more complicated. I'll take a sip of water here. There we go. Okay, so in the stomach, it's basically an expandable sac where your food will sit for a varied period of time. It depends on the complexity of the meal, how long it sits there. There is going to be motility, smooth muscle contractions will mix the food via peristalsis and segmental contractions. And then as the food is sort of mixed with the digestive or gastric juices that I'm going to talk about here in a second, small amounts of that will then be allocated to the small intestine over the next 30 minutes, 60 minutes, 90 minutes, depending on how much you're eating. There's a lot of secretions that happen here into the digestive tract here at the stomach. It's collectively called gastric juice, and it contains hydrochloric acid. We'll talk about how the stomach synthesizes and releases hydrochloric acid in a few slides. There's a bunch of enzymes, and as a category, they belong to a category of digestive enzymes called zymogens. Zymogens is a large category of digestive enzymes that are secreted in an inactive form. Because you got to remember here, the digestive system is in charge of digesting carbohydrates, lipids, and proteins. And these are the building blocks of your cells, especially lipids and proteins. And so if cells of your digestive system synthesized enzymes, digestive enzymes, in an active form, they would basically potentially start digesting themselves and kill themselves. That would be bad. And so a strategy that has evolved is that a lot of your enzymes for digestion are synthesized in an inactive form. And then once they're dumped into the digestive system, there are various methods present in the digestive system to then activate them. They're only activated when they are in the compartment where they need to work. And one example of that is pepsinogen being converted into pepsin in the stomach. And so pepsin is an enzyme that digests proteins in the stomach, starts the digestion of proteins in the stomach. When you see this suffix, sinogen, that is implying that this enzyme is in an inactive form. Pepsinogen is the inactive version, and then when you see an enzyme end in sin, then that is the active form. And so this is sort of the older naming convention for enzymes, before the whole ACE kind of thing. So we're going to see some older enzymes that don't have that ACE at the end. Sometimes enzymes end in sin or sinogen. So keep that in mind. When you see something that ends in sinogen or ogen, that's an inactive form. And then when it ends in sin, that's an active form. And we'll talk about here in a second how hydrochloric acid is what actually activates pepsinogen to pepsin in the stomach. There's also another fat digesting or lipid digesting enzyme in the stomach called gastric lipase. And then there's a lot of mucus that's going to be produced by the stomach lining, and it's full of bicarbonate to protect the lining of the stomach from this hydrochloric acid. Bicarbonate will neutralize acid as it starts to get close to the walls of the stomach. And so when your food, that's what I'm talking about over here, when your food that you eat mixes with this gastric juice, we then call it chyme. It's not just food anymore, it's food plus a bunch of secretions, so we call it chyme. So what's being digested in the stomach? Well, proteins for sure, because of the pepsin that's being released. The hydrochloric acid that's released into the stomach certainly does protect us against pathogens, but where it factors into protein digestion is that it denatures proteins. It's a very acidic environment in the stomach. pH 1, pH 2, very acidic. Causes proteins to unravel or denature. And so that exposes their peptide bonds to pepsin so that it can start breaking some of them. And then fats, again, very little will actually be digested here because fats have not been emulsified yet. That will happen later on in the small intestine when bile enters the scene. But a little bit of fat digestion can occur. Fats are kind of still globbed into big fat boulders at this point. They're getting away from the aqueous gastric juices. And so enzymes can nibble at their edges a little bit, but not a whole lot. And then what is absorbed through the lining of the stomach into the blood? Well, not any biomolecules, but aspirin, like pain relievers, can be absorbed across the stomach lining into the blood, which is great because if you're taking a pain reliever, you would like to relieve that pain as quickly as possible. Alcohol, alcoholic beverages, that kind of stuff, can absorb across the stomach lining very quickly and get into your blood, and you're dancing on the table with a lampshade on your head. Not a personal story, but you know, the cliche. So alcohol can certainly get into your blood and have an effect on your behavior quite quickly because it can absorb across the stomach lining. All right, so let's get into these gastric glands and how are enzymes and acid produced when you either see or smell food or food actually enters into the stomach. So here is sort of a cartoon drawing of your stomach, and remember that it's got these rugae, I forget exactly how it's pronounced, these sort of folds that can sort of expand the stomach in terms of size to accommodate meals of various sizes. So down inside these folds are these many of these gastric glands that you see here, which has a whole bunch of different kinds of cells, all sort of interspersed within a given gland. And this table is really great to sort of show you or point out these cells. And I wouldn't, Photoshop all of this out and make you identify these cells by color or shape or anything like that. But what I do want you to know is that up here towards the surface of these gastric glands and the neck region of these gastric glands, this is where the mucus is being produced. And that mucus is going to form a physical barrier between the lumen of the stomach and the epithelium. And it's full of bicarbonate, which buffers the gastric acid or neutralizes it to prevent damage to the stomach epithelium. And then as we go a little bit further down, now we get into the real workers of these gastric glands. The parietal cells, which make the gastric acid, the hydrochloric acid, which will kill bacteria that come in on our food, and it also activates pepsin from pepsinogen. It also denatures proteins, exposes their peptide bonds for pepsin to get in there and start cutting up. Also releases another substance called intrinsic factor, which is important for vitamin B12 absorption. Helps to permit that to be absorbed. We'll talk about these other green guys over here called enterochromaffin-like cells. They release a paracrine signal molecule that you've heard about before, histamine. We've talked about histamine in the immune system. We've talked about it in the respiratory system. And here's histamine working in the digestive system as well. And then chief cells are going to be the ones that release the two different enzymes. Releases pepsinogen, which once it's activated can digest proteins. And then gastric lipase, which as far as I know doesn't need to be activated, it will then digest fats. This is not a zymogen as far as I know. And then these D cells and G cells, we'll get to those as well. They are important for regulating the production of acid by parietal cells and enzymes by the chief cells. You want just the right amount of acid and enzymes produced for a given meal. And so when you have a small meal, you have a small amount of enzymes and acid made. If you have a large meal, then you have a large amount of acid and enzymes made. So these guys are involved in the regulation, the production of acid and enzymes. And the G cells release a paracrine signal molecule called gastrin, and it's the go signal. G cells, gastrin, go signal. D cells make or produce or release a signal molecule called somatostatin. It is the stop signal for acid and enzyme production. So I guess it's kind of convenient that these signal molecules are named gastrin, it is the go signal, and somatostatin, the stop signal. So this is a sort of step-by-step diagram, a story that you're going to want to be able to tell. And I highly recommend using process maps to learn these stories in Unit 6. I said it before on the reproductive lecture, like for learning the series of events that goes into meiosis, or this hormonal control of spermatogenesis, or the menstrual cycle. Use a process map to study those so that you can force yourself to try to tell the story. Well, process maps can also help with this sequence of events that happens in the gastric glands to result in acid and enzyme production in the stomach. When food either arrives in the stomach or when you see and smell that wonderful plate of buffalo wings on its way to your table when you're at Buffalo Wild Wings. I prefer Wingstop myself, but there's not a whole lot of Wingstops around here. So, yeah, I prefer Wingstop. That's okay. I digress. So here's the way the story begins. Remember, we always start with these yellow ovals. These are the stimuli, and we're not doing feedback loops, but you always want to start with a stimulus. That's what kicks off any sort of sequence of events. And so when food is When food is arriving in the stomach, it probably has amino acids and peptides in it. Well, you have little chemoreceptors in the gastric mucosa that will detect the arrival of these amino acids and short peptides. And that will activate sensory neurons, which don't go up to the medulla oblongata or the hypothalamus. No, they're part of the enteric nervous system. So these sensory neurons will just project to nearby G cells. And so anytime you see an arrowhead that has a sharp arrowhead here, that's an excitatory or stimulatory interaction. So these enteric sensory neurons will stimulate the G cells to release their signal molecule called gastrin. And that's a paracrine signal molecule. It just travels a very short distance to neighboring cells. The sight and smell of food is also transmitted via this black arrow to the G cells. And so that's transmitted via parasympathetic innervation to the enteric nervous system via the vagus nerve. This is one of the cranial nerves. So this is where the parasympathetic autonomic efferent pathways can also have a hand in stimulating the release of this signal molecule called gastrin, which, like I said, is the go signal for the rest of this. So now let's follow this. Gastrin then will just travel a very short distance to neighboring parietal cells. And parietal cells will be then stimulated to produce hydrochloric acid. Oopsies, that's on the next slide, which we're going to get to here in a second. Didn't mean to go there quite yet. There we go, let's go back to this. So parietal cells will release hydrochloric acid into the lumen of the stomach. That's where we're at over here. There's also vagus nerve input to the parietal cells as well. So either the sight and smell of food can fire this up or actual food entering into the stomach. Now there's also a sort of a third sort of backdoor way to stimulate these parietal cells. Gastrin will also trigger the ECL or enterochromaffin-like cells to release that other paracrine signal that I mentioned before called histamine. And so all of these three arrows here can stimulate the parietal cells to get to work producing hydrochloric acid. So the magnitude or how much hydrochloric acid is determined by how much of these signals are coming in to the parietal cells. They respond to how much stimulation they're getting. If you're getting lots of stimulation, it's a big meal, lots of food entering the stomach, you can certainly see and smell food all around you, well then yeah, there's going to be a lot of stimulation of parietal cells and you're going to make a lot of acid. If you just have like a little mini Snickers bar, well, you probably don't smell that. You do definitely see that. You might smell it a little bit. It enters into the stomach. It's just a small little mini Snickers bar. Well, there's only going to be minimal stimulation to the parietal cells here. And so they will produce only a minimal amount of hydrochloric acid. So this is how the magnitude of the meal can be linked to the magnitude of acid production in the stomach. Very cool. There are also chemoreceptors for these hydrogen ions. And so as acid is being dumped into the stomach, it will trigger these chemoreceptors, which activates enteric sensory neurons again to then stimulate the chief cells. Chief cells are the ones that actually release the enzymes, pepsinogen and also gastric lipase. You should draw in gastric lipase coming out of these as well. And if we were in lecture, I'd draw it in right now. But I don't know why they don't put it on here, but chief cells release both pepsinogen into the lumen of the stomach and gastric lipase. And gastric lipase is ready to go right out of the box. But pepsinogen needs to be converted into pepsin, and it's the hydrochloric acid that does it. That very acidic environment causes a change in structure for pepsinogen to become the active form pepsin in that very acidic environment, which did not exist inside these chief cells. These chief cells are 7.4-ish pH. And so the protein has a different shape when it's built in the chief cells than when it's in this super acidic environment. So that's how acid and enzymes are released into the stomach. And then we need to turn this off too, because you don't want to make like 6 gallons of acid and enzymes for a little mini Snickers bar. Well, how does this all get turned off? Well, there are D cells. This is the negative feedback pathway here. D cells will be stimulated when they sense sort of a threshold of hydrochloric acid. And when they detect a particular level of hydrochloric acid, they will release their paracrine signal molecule called somatostatin. And all of these dashed arrows with those little blunt endings there, these are all inhibitory commands or influences. So somatostatin, like I said before, is the stop signal. It will turn off the G cells, the enterochromaffin-like cells, the parietal cells, and the chief cells, if you follow all of those dashed lines. And so the whole system shuts down so that you only make a certain amount of acid and enzymes proportionate to the meal that you're eating under normal circumstances. So that's a tough story. And you may have to listen to that story a number of times until you can do a process map on your own. Push yourself to be able to do a process map of that on your own. Take all of these different pieces, everything that's in a yellow oval, a purple rectangle, a red diamond, etc. Take all of these terms and put them on like little Post-it note cards. Put them in the right order and then assemble a process map that has little action words next to each arrow. Okay? That will be a way that you can self-test. Make sure that you can tell this story before the final exam. All right, let's talk about those parietal cells in a little bit more detail. So the parietal cells here, these are in the walls of those gastric glands. This is the apical membrane over here. It faces the lumen of the stomach. This is the basolateral membrane over here. It faces the interstitial fluid and the capillaries that surround the stomach. And so how do these cells make hydrochloric acid and pump it into the lumen of the stomach? Great question. It all starts off with the same chemical reaction over here that we learned about in Unit 5. The conversion of water and carbon dioxide into hydrogen ions and bicarbonate ions. It's just written in a screwy way. And I don't know why the textbook does this. I don't think they're consciously trying to screw with you, but it is kind of screwing with you. But water and carbon dioxide go in, hydrogen ions and bicarbonate ions come out. And it's all being handled by that same enzyme again that we heard about before. Ca stands for carbonic anhydrase. And so this is how a surplus of hydrogen ions are produced. And so let's follow them into the lumen of the stomach. They're going to get pumped through these carrier proteins right here. And because you see an ATP, you should think, oh, that's a carrier protein doing primary active transport using ATP as its energy source because it's pumping hydrogen ions against their concentration gradient, and also potassium is going against its concentration gradient that we've learned about throughout the entire course. Hydrogen ions are going against because there's lots of hydrogen ions out here. It's very acidic, so obviously there's tons and tons of hydrogen ions out here. These are called hydrogen ion pumps, or also called proton pumps, because a hydrogen ion is just a proton, technically. A hydrogen atom, like from the periodic table, hydrogen atom has one proton and one electron. And so if it loses that one electron and becomes positively charged, it must then therefore just be a proton. So that's why these are also called proton pumps in some references. And then chloride ions will get pulled out through these little leak channels right here because of the very positively charged environment out here, pulling chloride against its concentration gradient. It's basically going down an electrochemical gradient, but this is still facilitated diffusion going through a channel. And then you see these potassium ions just sort of basically circulating perpetually through this carrier protein and then back out through a leak channel, through this carrier protein, back out through that leak channel. So that's how hydrochloric acid is pumped into the stomach. Well, what happens with the bicarbonate? Well, let's go over here. Bicarbonate needs to be exported from these cells. And so it will go through these carrier proteins here. It's doing the chloride shift that we learned about in Unit 5. These carrier proteins are doing secondary active transport. Chloride is going down its concentration gradient, and bicarbonate is going against its concentration gradient. So these are doing secondary active transport. And that bicarbonate will join the blood, and that's what I'm talking about over here. Bicarbonate enters the blood, and that adds pH buffering capacity to the blood, which will be important later on in the story. So remember that bicarbonate was added to the blood to work as a pH buffer at this point. And then just a little bit more on these hydrogen ion pumps or proton pumps. You maybe have heard of drugs called proton pump inhibitors, like Prilosec, Nexium, Protonix. These drugs will block, to some extent, these proton pumps right here on the parietal cells. And so they are used, the conditions that they're prescribed for, or people buy them for, you can buy them over the counter too, they are used for people that have chronic heartburn, chronic overproduction of acid in the stomach. It will acid reflux up into the esophagus and causes irritation, burning sensation behind the heart. We call it heartburn. It also leads to what's called GERD. That's an acronym, GERD. Stands for gastroesophageal reflux disease. Gastroesophageal is one word, reflux, and then disease, GERD, is the more formal name for the dangers of too much acid refluxing back up into the esophagus, causing heartburn. It can be dangerous to have too much acid irritating the esophagus. It can cause cellular damage. It can be a risk factor for esophageal cancer. So if you or you know somebody that suffers from chronic heartburn, chronic acid reflux, it is something to be taken seriously because it could lead to esophageal cancer. And then the last little thing that I'll say before we leave the stomach is about the production of mucus. Remember these mucus surface cells right here, and then a little bit further down, these mucus neck cells produce the mucus plus the bicarbonate that protects the gastric sort of epithelial cells from damage from this very, very acidic gastric juice around pH 2. And so if any stray hydrogen ions get into this mucus, they're immediately neutralized by bicarbonate. So that's how your cells of your stomach can stay safe from this very hazardous acidic environment produced by the gastric juices. And I guess that's all I want to say about the stomach at that point. So let's move on to the small intestine. I gotta have a sip of water. Okay. How am I doing on time? I'm doing good on time. Okay, so into the small intestine now. So this is where the vast majority of your chemical digestion of food occurs. Lots of enzymes being released into the small intestine. The vast majority of your absorption of biomolecules into the blood. So we're going to spend a good amount of time here in the small intestine. So still lots of peristalsis to move food from one place to the next, and also segmental contractions to keep everything mixed up and in contact with the absorptive epithelial border of the small intestine. The secretions that are going to be produced, finally now we get bile coming from the liver. It was synthesized in the liver and by way of the gallbladder. And so the gallbladder is a little accessory little sac that hangs off the bottom of your liver. And, well, it's not technically connected to the liver, but it's sort of located underneath your liver. And the idea is that the bile is synthesized in the liver and temporarily stored in the gallbladder before hormonal cues will cause the gallbladder to contract and squirt that bile into the small intestine. That will be important for fat digestion, lipid digestion we'll get to later on. There's also lots of pancreatic secretions called pancreatic juice. Lots more zymogens, proenzymes, that kind of thing. So zymogens, again, are inactive enzymes. They are activated upon entering into the small intestine. And then the pancreas also makes, inside this pancreatic juice is a lot of bicarbonate. And so this bicarbonate is going to be critical to neutralize the acidic chyme coming from the stomach, because the enzymes that work in the small intestine don't like a pH 1 or pH 2 superacidic environment. They can't operate. That's outside their optimum pH. They can't operate under those conditions. So enzymes in the small intestine actually prefer a more of a basic or alkaline pH. And then we're not going to talk so much about the hormones, but there are hormones, like I already said, that control the squeezing of the gallbladder to remove or to release bile, that kind of thing. So this is where proteins are broken down. Lots of enzymes that are breaking these huge, massive proteins down into either single amino acids or short polymers, dipeptides, tripeptides, oligopeptides. Remember, oligo means a few. So very short fragments. And then carbohydrates are further broken down by pancreatic amylase. Remember, amylase was released by the salivary glands, but that enzyme denatures in the acidic stomach. And so this is another boost of amylase from the pancreas. Pancreatic amylase continues the breakdown of complex carbohydrates. And then there are di and trisaccharidases. These are enzymes that break down di and trisaccharides into monosaccharides. All carbohydrates need to be monosaccharides before they can be absorbed. So glucose, galactose, and fructose are the three monosaccharides. Fats are now going to be digested in earnest at this point now, because bile will finally emulsify the fats into smaller droplets, allows them to finally mix with the aqueous juices and secretions of the digestive system. They are not just hanging out off to the side in big fat globs. They can finally mix with the enzymes that are meant to break them down, like lipase and colipase break down triglycerides into monoglycerides and free fatty acids. And so that's what we're going to be absorbing: monosaccharides, amino acids and short peptides, monoglycerides, fatty acids, and also cholesterol. This is where we're going to spend the majority of our time, going through these processes. I'm not going to talk so much about water and electrolyte absorption. It's not as interesting, as these solutes and electrolytes are absorbed into the blood. Well, water is going to follow. Water always follows solutes by osmosis. So I'm not really going to talk much about these two. I'm going to talk about these important biomolecules for fuel around the body. So this is a figure from your textbook showing these pancreatic secretions coming from the pancreatic duct into the lumen of the small intestine. So what do these pancreatic secretions include? Well, they include a lot of bicarbonate, because we're going to need that to neutralize the acidic chyme coming from the stomach, make a more alkaline pH for the enzymes of the small intestine. We got to keep it all sloshy and soupy, of course. So mucus and isotonic saline are a big component of these pancreatic secretions as well. Bile, like I said before, is produced and secreted by the liver, and it's temporarily stored in the gallbladder. Some people have gallbladder issues, gallstones, something like that causes a problem with that organ. It may need to be removed. You don't have to have a functioning gallbladder to survive. If you have your gallbladder removed, the body will just adapt and the liver will just produce bile on demand rather than sort of making it ahead of time and storing in the gallbladder. It just makes it on demand. It's totally fine. And then a lot of new pancreatic enzymes. Again, they're secreted in an inactive state. They are part of that category called zymogens, which are shown here in sort of orange or yellowish kind of color. They're activated when they get into the small intestine. The why is so that they don't digest the cells that produce them, because these are enzymes that are involved in fat and protein digestion. How are they activated? Well, I'm going to show you that on the next slide. So this is where I'm saying, finally, why these enzymes are produced in an inactive form. They're activated in the small intestine where they're actually going to do their work. So how do they get activated? How do these pancreatic zymogens get activated? Well, that story is being shown over here. So these pancreatic secretions come in, and along the walls of the small intestine, these intestinal mucosa cells, remember they have these little finger-like extensions called villi. Well, this is also called the brush border because it has kind of a brush-like or comb-like appearance. And so there are enzymes that are actually mounted on the exterior walls of these villi and microvilli. And so those enzymes that are mounted on the exterior walls, exterior membrane, sorry, I shouldn't say wall, the exterior plasma membrane of these intestinal mucosa cells are called brush border enzymes. And one of those brush border enzymes is called enteropeptidase. Enteropeptidase would be mounted on the brush border right here, on the exterior plasma membrane of these small intestine epithelial cells. And so when these pancreatic secretions come in, these pancreatic secretions come in, these brush border enzymes like enteropeptidase will activate trypsinogen into trypsin. And then once trypsin is activated, it will activate all the rest of these zymogens. It converts chymotrypsinogen into chymotrypsin. And then the rest of these all end in ACE. And so when a zymogen ends in ACE, its inactive form has a pro at the beginning. See? Procarboxypeptidase is converted into carboxypeptidase. You just drop the pro prefix. Procolipase is converted into the active form colipase. Prophospholipase is converted into phospholipase. And so that's how these pancreatic zymogens are activated. It's like a domino effect. Starts with the brush border enzymes activating trypsin, and then we're on our way. This cell diagram is showing you how cells of the pancreatic duct and in the duodenum, the first section of the small intestine, make the bicarbonate. How do they release bicarbonate to neutralize the acidic chyme in the lumen of the small intestine? Well, it's just like a parietal cell that we saw up in the stomach, but it's flipped 180 degrees. It's flipped such that now bicarbonate is being released into the lumen of the digestive tract. And look, hydrogen ions are going the other way this time, into the interstitial fluid, and they would join the blood. But don't worry about these hydrogen ions being pumped into the blood, because remember, there's extra bicarbonate buffer was put into the blood preemptively by the stomach, by the parietal cells. So it's almost like the parietal cells in the stomach are working with these cells that are in the small intestine and in the pancreatic duct. They're working together where there was preemptive loading of bicarbonate into the blood to deal with these hydrogen ions later on. But it all starts off with the same chemical reaction again drawn in a kind of a screwy way. But water plus carbon dioxide ultimately makes hydrogen ions and bicarbonate ions. And in this case, now the chloride shift carrier proteins are on the apical membrane. They pump bicarbonate out, and chloride is going down its concentration gradient here, and so that is secondary active transport. And the chloride just circulates back around and around and around as needed. Can go get pulled back out through these leak channels. The hydrogen ions get pumped out over here using secondary active transport through that carrier protein right there, with sodium going down its concentration gradient and hydrogen ions being pumped out. So that's doing secondary active transport as well. And then the rest of these down here, sodium potassium pump, the NKCC cotransporter that we saw in the urinary system, these are just maintaining the ionic disequilibrium around these cells. And that potassium leak channel, just maintaining the ionic disequilibrium around these cells to keep these carrier proteins working properly. Okay? So that's another cell diagram. Hopefully by this point you've gotten a little bit more seasoned at looking at a cell diagram and analyzing it. The upper portion of this is what really allows this cell to make bicarbonate. These portions down here just are maintaining the ionic disequilibrium around this cell. Okay? So let's talk about the digestion of carbohydrates here in the small intestine. So by this time, glucose polymers have gone through the mouth where a little bit of digestion occurred, very brief, by salivary amylase. Carbohydrate digestion goes on pause in the stomach because there's no carbohydrate digesting enzymes released by the stomach, and amylase gets denatured in the stomach's acidic conditions. So now we get another burst, another release of amylase coming from the pancreas now. And it continues the breakdown of starch and glycogen, which are massive, massive, huge polymers. They're not this long. They're massive, like thousands or tens of thousands of glucose monomers linked together. So amylase continues to chew them down to the level of disaccharides, like maltose, sucrose, and lactose. And then there are disaccaridases that are attached to the apical membrane, the brush border of the intestinal mucosa cells. So these enzymes here, maltase, sucrase, and lactase, are just attached to the brush border of the small intestine. And so maltase converts maltose into 2-glucose monomers. Sucrase cuts sucrose into 1-glucose and 1-fructose. And then lactase tears apart lactose into 1-glucose and 1-galactose. And so by this time now, we've got all of these monosaccharides ready to be absorbed. And that's what's being shown on this figure right here. So this is showing you how these various monosaccharides are transported from the lumen of the small intestine, trans-epithelially, if that's even a word, across the apical membrane here and the basolateral membrane here to get into the interstitial fluid and then join the blood, the capillaries that surround the small intestine. And then this is the pathway that fructose has to follow. Glucose and galactose have this more of a hexagon sort of shape to them. They're very, very similar in structure, so they can use the same pathway. Fructose is more of a pentagon-shaped ring molecule. And so because of its different shape, it has to follow a different pathway over here. So glucose and galactose can be pumped across the apical membrane in co-transport with sodium. This is the sodium glucose transporter, or sodium glucose co-transporter, that we learned about way earlier in the course in Unit 2, but we saw again just recently in Unit 5 at the proximal convoluted tubule of the nephron. And so this is a carrier protein doing secondary active transport with sodium as the power source, pumping glucose and galactose into these cells. And then they will leave by facilitated diffusion via GLUT2s. That transporter right there is the old GLUT2s that we learned about in Lab 13 when we were talking about insulin and glucagon. So these GLUT2s we learned in Lab 13 just do facilitated diffusion. And as far as I know, they haven't changed. Now fructose, because of its different structure, it enters via a carrier protein called a GLUT5. And the way that I remember it is that fructose is a pentagon-shaped ring, which has 5 sides. Enters via a GLUT5. That's how I remember it. And it leaves via the GLUT2s. Obviously, the GLUT2s are not as selective in what kinds of monosaccharides they let through, because this is a GLUT2 right there, and that's a GLUT2 right there. And so ultimately, these glucose and galactose monomers get transported or absorbed into the blood, and also these fructose monomers get absorbed into the blood. And so what happens to them from there? Well, let's go on to the next slide. So these capillaries then flow into the hepatic portal vein, which transports all these monosaccharides to the liver. Well, what happens to glucose? Well, let's go down here. Glucose, we know from Lab 13, is stored as glycogen in the liver or in muscle cells. Or it can be converted into fat, into triglycerides in adipose tissue. Or some glucose that you eat just stays in the blood and it's delivered to cells for immediate use in cellular respiration. But what about these other two, fructose and galactose? We didn't learn any metabolic pathways for them. Well, they are converted into glucose in the liver. Fructose can also be converted into fat in adipocytes. This is one of the dangers of foods that have high levels of fructose, like higher than a piece of fruit. Fructose is the main sugar in fruit, and there's nothing wrong with eating fruit with fructose in it. But a lot of processed foods that we'll talk more about in our last lecture of the semester have super high levels of fructose, high fructose corn syrup. And so the more of those processed foods that you eat with lots of fructose, the more fructose is converted into fat in your body, and that can lead to obesity. We'll pick up that topic in the last lecture here coming up in the next couple days. All right, moving on to protein digestion in the small intestine. Proteins are massive, massive, massive molecules. A lot of the proteins that we ingest, especially when you eat animal products, meat, that's muscle tissue, right? We know muscle has lots and lots of protein, and those proteins are huge. Like Titan, for example, the biggest protein as far as I know that is known. Huge. It's like tens of thousands or 50,000 plus amino acids in one protein. So proteins are massive, massive strings of amino acids. And so that's why we need a lot of enzymes for digesting them. This diagram does not do a protein justice. They are massive, massive molecules, 10,000, 50,000 amino acids long. And so holding those amino acids together in this figure here, this is an amino acid right here. That blue square right there is an amino acid. That's another one, that's another one, that's another one, etc. These lines, these horizontal lines, those are the peptide bonds. Those need to be clipped. They need to be broken by these enzymes. And so there is a category of protein digesting enzymes called endopeptidases. Endopeptidases is a category of enzymes. It includes pepsin from the stomach, trypsin and chymotrypsin in the small intestine. The sinners, pepsin, trypsin, and chymotrypsin are all sinners. They all end in sin. That's how I remember that they are endopeptidases, or part of the same category. These enzymes grab onto two adjacent amino acids at their binding site, and they break the peptide bond in between it. They break that bond right there. Or they may grab onto these two amino acids and clip that peptide bond right there. So basically, they digest proteins by attacking the interior peptide bonds. There is another category of protein digesting enzymes called exopeptidases. Exopeptidases attack amino acids at the ends of a long chain of amino acids. Exo means outside, endo means inside. And so aminopeptidase will identify this amine functional group and it will clip the peptide bond that is closest. Carboxypeptidase will identify this carboxyl functional group and clip the closest peptide bond. And so what we've got going on here, and to answer my question out here, why do you think there are so many enzymes involved in protein digestion? Because as molecules, they're freaking huge, and we eat a lot of protein. And so to efficiently break down these massive molecules, and a lot of them, we need a group of endopeptidases breaking peptide bonds in the interior, and we need a category of enzymes called exopeptidases, which clip off single amino acids from the ends of these proteins that we eat. It's a team effort. It makes things much more efficient. That's the idea, to have all of these protein digesting enzymes. And so the job of these endo and exopeptidases is to take these massive proteins up here and turn them into smaller fragments, either single amino acids or di and tripeptide level, or oligopeptides, small peptides. These are too big to fit through carrier proteins. These can fit through carrier proteins. So we've got 3 lanes or 3 options for getting protein subcomponents or molecules into the blood. And so let's go through this one right here. This is our old friend transcytosis going on here for the oligopeptides. They're too big to fit through carrier proteins, so they need to go on the apical membrane, into these cells of the intestinal mucosa here, into them via endocytosis, into a vesicle. That vesicle is going to be trafficked across the cytoskeleton by motor proteins, dragging the vesicle from one place to another. And then this vesicle will dock with the basolateral membrane and undergo exocytosis. And then these fragments are small enough to get into the capillaries. And they'll be on their way to the liver to either be converted or used for other purposes. They can be delivered to the liver to make glucose. And remember, the conversion of amino acids into glucose is called gluconeogenesis. It's a way of making glucose from other biomolecules. Let's go to this one here now. This is the single amino acids. They're going to go through carrier proteins on the apical membrane and the basolateral membrane in co-transport with sodium. And so this is secondary active transport again. Allows these single amino acids to be pumped in and then pumped out. And then they join the blood. And then for di and tripeptides here, these are carrier proteins here and here, but it's co-transport with hydrogen ions in this case. And so these hydrogen ions can be used as a concentration gradient to pump di and tripeptides into these cells. And then they leave via co-transport with hydrogen ions again. And so those are carrier proteins doing secondary active transport. They're just hydrogen ion dependent secondary active transport. There is kind of a surplus of hydrogen ions around at this point because we've got a lot of amino acids. These are acidic molecules. Amino acids are not just cleverly named that. Amino acids can release hydrogen ions that can be used as a concentration gradient. There's also fatty acids being digested in this area. So fatty acids can also release hydrogen ions. That's where this hydrogen ion gradient comes from to power this. So that's the story of how we absorb either oligopeptides, di and tripeptides, or single amino acids into the blood. And they are then delivered all around the body as building products, building materials for making proteins of our own, but also could be delivered to the liver where they can be converted into glucose, which of course is a super important sugar for cellular respiration. All right, moving on to lipid or fat digestion. I've been using these terms kind of interchangeably. They basically are interchangeable. And so lipid and fat digestion did begin in the mouth and continued in the stomach. There is some lipase activity in those earlier compartments. But the problem is that fats didn't mix with the aqueous saliva or the aqueous water-based gastric juice, because fats are nonpolar. Nonpolar molecules don't mix well with water. Saliva is a water-based fluid. Your gastric secretions are a water-based fluid. Oils and water don't mix. And so, or fats and water don't mix. And so up till this point, the fats that you eat have been globbed together into big fat boulders, is the way that I like to call it or mention it. But now we have a sort of a secretion called bile. It's an emulsifier. Bile is an amphipathic molecule. Amphipathic. And what that means, amphipathic is a descriptor for a molecule that has a polar end and a nonpolar end. And so what happens when bile is released from the gallbladder, it was originally synthesized in the liver, but once it's released from the gallbladder into the small intestine, it will emulsify big, huge fat globules of triglycerides into smaller droplets. And so what happens is that the nonpolar end will insert itself into the fat, and the polar end will face out towards the water. And so it emulsifies the fat. It allows it to break into smaller bundles, smaller packets, and now they will be able to dissolve in the blue area here, which is the aqueous environment of your small intestine. And now enzymes can get at them in large amounts, at least. And so now these triglycerides and such, the fats that are in these little smaller emulsified packets here, can now be attacked by enzymes like lipase and colipase. So I tell you a little bit more about what they do. Lipase and colipase work together. Colipase will displace some of the bile, kind of breaks off some of the bile from the droplets. Allows lipase to then get in there and start breaking the ester bonds that are holding these fatty acids to glycerol right here. And so what comes out of this process are monoglycerides and free fatty acids. These can now be absorbed into across the intestinal wall. Phospholipase is an enzyme that's involved in breaking down all the phospholipids that we eat. If you eat plant or animal cells, you're eating a lot of phospholipids from their plasma membranes. And so that will break those down into free fatty acids. And so now at this point, we've got the story now of how these lipids are absorbed across the wall of the intestine into, not into the blood this time, into the lymphatic vessels, the lacteals that are here around the small intestine. So let's go through this story. Some nice sort of bullet points down here from our textbook. But the idea, we start off with large fat droplets that have come from the stomach. They have not been digested hardly at all because they are very, very nonpolar. They don't mix well with the aqueous solutions of the mouth and the stomach. And so here we've got bile coming from the gallbladder, is now released into the small intestine. It will emulsify these big, huge fat droplets and makes them into smaller globules. And now they can actually dissolve in the aqueous environment here in the lumen of the small intestine. And then here at point 2, this is where those enzymes, pancreatic lipase and colipase, actually break down the big, huge triglycerides into monoglycerides and fatty acids, which are stored in smaller, very small fat little droplets called micelles. These micelles contain cholesterol, that's what that 3B is, cholesterol, and monoglycerides here and free fatty acids. Now these are very nonpolar, so they can go right across the plasma membrane. Remember, we've talked about nonpolar molecules being able to freely go across plasma membranes since almost day 1 in this class. And so they are nonpolar. This is just, this here, these arrows here are just simple diffusion. See, you don't see any carrier proteins or channels or anything. So that just must be simple diffusion. We'll come back to them here in a second. Cholesterol, 3B right here, Cholesterol is another one of these amphipathic molecules. Amphipathic is a molecule that has a polar end and a nonpolar end. So it can't go freely across the plasma membrane here. It does go through a carrier protein, which they made very, very small here. I don't know why. That little blue egg shape right there. That's a carrier protein. It gets the super sexy name, NPC1L1 transporter. Who the hell named this? Come on, be a little bit more creative. Call it the cholesterol transporter or something like that. No, it's called the NPC1L1 transporter. Well, why am I highlighting that? Well, the reason why I'm highlighting that is because we've talked about cholesterol before. Cholesterol is an important part of your diet. You do need cholesterol in your body in order to synthesize steroid hormones. We've talked about steroid hormones a lot lately, especially in the reproductive lecture. So you need a supply of cholesterol to synthesize either estrogens or progesterone if you are a female, or the androgens if you're a male. You also need cholesterol for plasma membrane repair, growth, that kind of thing, because cholesterol is part of the plasma membrane. But the problem for a lot of people is that their diet contains way more cholesterol than their body really needs. And so then cholesterol goes from being a good thing to being a villain and being a risk factor for cardiovascular disease. So you're about to go out in the world, off to your life, and hopefully to a very fruitful career, maybe in healthcare. You're going to see a lot of people that have cardiovascular disease and cholesterol management problems. You are. And so I want to introduce you to some drugs right now that are involved in managing cholesterol in people that can't manage it with dietary changes. So there is a drug called Zetia. Maybe you've heard of this on TV. I've seen it advertised on TV. Zetia. That decreases the action of this transporter right there. And so it can be used to treat patients with high cholesterol and a high risk of cardiovascular disease. So there is a drug that's now on the market that can help to block that carrier protein right there and keep cholesterol from getting into the body. So that's one kind of drug that is used to manage cholesterol. Now I want to contrast that with what I'm talking about over here. Is maybe you've heard of a category of drugs called statins. They're actually the most prescribed drug on the planet. And they have a lot of kind of nasty side effects, but they're also used to help people that are having trouble managing their blood cholesterol levels. And so those drugs are called Lipitor, Crestor, Zocor. Maybe you've heard of these too. I see them advertised on TV. But they're part of a different category of drugs called statins. They don't work here. They block the production of cholesterol in the liver. So they block your body's natural production of cholesterol, which can also help people to fight heart disease. So like I said, you're hopefully going to go off to a very fruitful career, and you're going to see a lot of people with cardiovascular disease and cholesterol issues. So I at least want to teach you about the two mechanisms by which drugs can help to fight high cholesterol in human beings. So there you go. Okay, so let's pick up this story from there. So now at this point, what we've got is we've got fatty acids and monoglycerides getting into this cell via simple diffusion. And cholesterol coming in. As far as I know, this is primary active transport, but it's not very clear in the textbook. But cholesterol is entering in as well. So what happens at that point? Well, these free fatty acids and monoglycerides will go through the smooth endoplasmic reticulum of these cells of the small intestine and will be reassembled into triglycerides, which is pretty crazy. They just got broken down from triglycerides. And then these cells resynthesize them into triglycerides. It's weird. Cholesterol and triglycerides and proteins that are being absorbed by these cells as well through the mechanisms that we just covered a couple slides ago, will be combined together, built into a big, huge particle called a chylomicron. And these chylomicrons are so huge, they're too big to leave via a carrier protein. They're huge particles. And so chylomicrons need to then go through the Golgi apparatus and get packaged into vesicles and released by exocytosis. And they're too big to go through the fenestrated capillaries here and join the blood supply. But they can go through these larger openings, these larger ducts, these one-way ducts, into the lacteals, which are the lymphatic vessels that surround the small intestines. They get a special name. They're called lacteals. And so these chylomicrons are too big to join the blood directly. They go into the lymphatic system. But they will be dumped into the cardiovascular system at the vena cava. Remember, all the lymph fluid eventually rejoins the blood at the vena cava. And then the body will process and store these fats in your adipose tissue. They'll be separated from the protein component later on down the road. Okay, so that's kind of the weird, wild journey from big fat boulders over here to these chylomicrons going through the lymphatic vessels to get access to the blood. All right, process map maybe again might be a good way to learn this. What goes on then in the large intestine? Good, I'm doing good on time. So what is happening? That's the end of the small intestine. It's a lot to learn. Definitely spend the majority of your studying time on the stomach and the small intestine. The large intestine, I don't have much to say. Like I said very early in this talk, the large intestine is almost like kind of a victory lap for your food. Almost everything has been absorbed at this point. So it's only just fine-tuning the composition of the poo, taking in as much nutrients as you possibly can before you let it go out of the body. So there is going to be continued peristalsis to move the food, the chyme, through the large intestine segments, the ascending colon, transverse colon, descending colon. Segmental contractions will continue to mix it with the mucus, keep it kind of loosey-goosey in that area. You don't want an obstruction or anything like that. You don't digest or you don't produce any more digestive enzymes. You don't. But you have a lot of symbiotic bacteria, symbiotic microorganisms that live in your large intestine. And we're learning more and more about the health benefits of keeping those little microorganisms happy and satisfied down there. So keep your eye on this topic maybe in the future, or if you still need to take microbiology, as far as I know, they talk a little bit more about this microbiome that lives in your large intestine. They're symbiotic. They live in symbiosis with you. And so they are able to break down the last remaining proteins and polysaccharides that you can't digest with your own enzymes. So you give them a nice, warm, moist place to live, and you also feed them with proteins and polysaccharides that you can't do anything with. And they pay you back, they pay you rent, basically, by synthesizing important vitamins that you need to survive or be healthy. And so what's being absorbed in the large intestine? Well, it's sort of finishing off the water and electrolyte absorption that started in the small intestine. But these vitamins that these microorganisms pay you back, pay you rent for living in your large intestine, like the B vitamins, the K vitamins, are super important to your health. And that's where they are, of course, then absorbed into the blood. And then you store the feces until you find a socially appropriate place to let it go. The defecation reflex is triggered by distension of the rectum, blah, blah, blah. That's all I'm going to say about that. And then it's out of the body, and that's the end of our journey at this point. So one slide on the large intestine, but as you go off into the world, maybe to a microbiology class or just out into the world, I would keep an eye on us learning more and more about these microorganisms in the large intestine and how they do seem to play a very critical role in your overall health. It's a very interesting topic, and I've tried to read a little bit more on it, but there's not a whole lot of really solid information on it at this point. So keep your eye on these microorganisms. We might learn a lot more about them in the near future and how to keep them happy and how they keep us healthy. And so what kind of questions will you will you see on the final exam about the material that I've just covered very quickly? I know, very quickly. Good thing it's a video. You can go back and watch it again. But what kind of questions are you going to receive on the final exam on this material? Well, you're going to need to know what happens in each of these different compartments. Know where biomolecules are digested and absorbed. So when you are asked questions like this, you're going to need to, answer questions like carbohydrate digestion occurs here. Occurs just means that it has enzymes in order to occur. Or I may ask you where, say, a biomolecule category like protein digestion begins here. So make sure you read the questions very closely. Am I asking you where the process begins, or where does it just occur? Where does it just happen? And so I'll let you do these little concept check questions on your own, choosing from this list. And if you want to check your answers, you're welcome to take a picture of this slide and send it over to me, and I will evaluate your answers. And then other kinds of questions that I might show you is you're going to want to know the nuts and bolts of digestion and absorption, especially in the stomach and in the small intestine. Those are the most important places. That's where we spent the vast majority of our time in this lecture. So choosing from this list right here of these various enzymes, or maybe an emulsifier here, hopefully you'll be able to answer these kinds of questions. And these are choose all that apply, so there may be more than one answer to these questions. So make sure when you answer the question, evaluate all of these answers. You won't see those kinds of questions on the final exam. I don't do choose all that apply very much anymore. But as far as I recall, those are not on the final exam. So it'll be pretty straightforward. All right, so that's it. Yep, technological meltdowns required me to do this lecture on the side, all by myself here. Faster than I normally would do it in class, for sure. Yeah, hour and a half. Listen to this lecture. Take good notes. I didn't get to write anything on any of these slides, so you're going to have to pick up things that I say and take your own notes without my little cues. And if you have any questions on this sort of stuff, please don't hesitate to send me an email. Okay, we're close to the end. It's critical time right now. So make sure that you are not shy about sending me questions, especially because this lecture was given, without you being able to ask questions along the way. Okay? All right, so that's it. Good luck. Enjoy. Talk to you again later.